This is Mrs. Vance again. It is now time for Meiosis 2, which is Unit 9 Notes, Part 3, about the second division of meiosis. Remember, in the last session we talked about the first division, the fact that we're separating the homologous chromosomes from each other. We had crossing over, synapsis, forming the tetrad, and the crossing over that occurred in prophase 1 of meiosis, and all the way through telophase 1, where we ended up with two daughter cells, each of which is haploid, but with duplicated chromosomes. So now we're going to pick up from there and talk about the next part, which is meiosis 2. So the big thing to remember, again, what we're doing is reducing the chromosome number from diploid to haploid. And we've already got haploid cells, but the haploid cells that we have duplicated chromosomes. So we still have twice as much DNA as we need for our gametes to have. So meiosis 2 is going to follow directly after meiosis 1. There's not an interphase in between. There's no chromosome duplication or anything like that. We're going to go straight from telophase 1, cytokinesis, and then go right into meiosis 2. And each one of those haploid products is going to enter the meiosis 2. And the first stage, of course, is prophase. Okay, so prophase 2, like we saw in mitosis, like we saw in meiosis 1, the chromosomes coil up and become compact. If, in fact, they uncoiled after telophase 1, that depends on the species. Different species do it slightly differently. But basically, if the nuclear membrane reformed, then it breaks up again, and we end up with something that looks like what we saw and what we see at the end of prophase, or in prophase of mitosis and meiosis 1. And so we're going to see the sister chromatids are going to, here we have the two daughter cells, each of which is haploid with duplicated chromosomes. The spindle is going to form again, like we saw before. This time spindle fibers from both ends will attach to each centromere, like we see in mitosis. In anaphase, the centromere is now going to split, and the sister chromatids, now separate chromosomes, will be pulled apart, and then we'll end up with telophase 2 and cytokinesis, like we saw before. But this time, our daughter cells will only have one copy of each chromosome, rather than the duplicated homologous chromosomes we had at the end of prophase, at the end of meiosis 1. All right, here we have some lily cells that are undergoing meiosis. You can see here the two daughter cells that formed here, and here's the second pair of daughter cells that are forming in this from this one, just to show you that you can actually see it under the microscope like that. Okay, so in metaphase 2, we have our duplicated chromosomes lining up at the cell equator, like we saw in mitosis. In anaphase 2, the sister chromatids are going to separate, and chromosomes will be pulled toward opposite poles of the cell, like we saw in mitosis. In telophase 2, once the chromosomes reach the poles, the nuclear envelope forms around each set. The chromosomes decondense back into their state of their noodle-like state of chromatin, and we have now 4 haploid cells after cytokinesis that are produced from that one parent cell that we started with at the beginning. The big thing to remember is that there are similarities definitely between mitosis and meiosis, but there are definite differences between them as well. They both begin with a diploid parent cell, and that the chromosomes have duplicated in during the previous interphase. But the end products are very different. In mitosis, we have 2 genetically identical daughter cells that are diploid, and they're somatic cells. That means they're body cells. Whereas meiosis produces 4 genetically different, genetically unique haploid gametes. And again, remember, meiosis only occurs in the gonads, in the sex organs, sperms, the ovaries and the testes, to produce the egg cells and sperm cells that are needed for reproduction. Here we have a pictorial comparison between the two processes, and this picture is in your book in chapter 8. Okay, so you can see in mitosis you have the duplicated chromosomes that are made of 2 sister chromatids here, okay, coming from the parent cell. In prophase 1 of meiosis 1, we have the duplicated chromosomes as well. However, the tetrad forms and crossing over occurs during prophase 1. It didn't happen in mitosis. In metaphase 1, the tetrads line up at the metaphase plate, whereas in metaphase of mitosis, the chromosomes align separately at the metaphase plate. In meiosis, we're going to separate the homologous chromosomes from each other during anaphase 1, whereas in anaphase of mitosis, we're separating the chromatids from each other. We end up with 2 diploid daughter cells here at the end of mitosis, whereas at meiosis 1, we have 2 haploid daughter cells with duplicated chromosomes that then have to undergo another set of divisions to produce the 4 haploid daughter cells, each of which, because of crossing over, is genetically unique from the other ones. Now, there are a lot of things that lead to the variation in genetics that occurs in members of a species, okay? One way that occurs is because of the variation in gametes from the independent orientation of chromosomes. In other words, they don't line up in order 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. They might be 1, 3, 5, 6, 2, 7, 8, and 10, and then 9, or something else entirely different. And so the way they line up is independent of each other. The way they line up in terms of the maternal and paternal chromosomes facing toward one end or the other is also very independent of each other. And then the other thing that we have that adds to genetic variation is random fertilization. That means you don't know which particular combination of chromosomes is going to be in any particular sperm or any particular egg. And so the fact that those 2 random cells come in contact with each other and make a new organism is totally independent. Okay? So remember, each pair of chromosomes independently aligns at the cell equator. There is an equal probability of the maternal or paternal chromosome facing one pole or the other. Okay? And you can calculate the actual possible combinations for the gametes, for the chromosomes, is 2 to the n power, where n is equal to the haploid number of chromosomes. And so in the case of humans, that would be 2 to the 23rd power. That gives us something like 20 trillion different combinations in humans. So lots of different combinations that you can have there of those chromosomes. Random fertilization, again, refers to the fact that the combination of each unique sperm with each unique egg increases that genetic variability. So if I have in my egg cells 20 quadrillion different possible combinations, and you have the same thing for the sperm cell, 20 quadrillion different combinations, you see that you've got a lot of variability there. It's definitely random in terms of which particular chromosomes end up in the zygote at the end. So here's just a pictorial representation of how we can see the different possibilities, the independent assortment here. Okay, we have 2 equally probable arrangements of chromosomes at metaphase 1. Dad's chromosome going one direction, mom's going the other in these 2 chromosomes, or we could have dad's number 1 and mom's number 2 going one direction, and mom's number 1 and dad's number 2 going the other direction. Okay, and so that would give us different combinations that you have. And so that's just compounded by having, when you have more, the more chromosomes you have, the more possibilities they are. And then when you add crossing over into that, that adds even more possible combinations. So you have a lot of different things going on there. Okay, we're going to stop here. There is a Amoeba Sisters video. Rather than playing it inside this video, I'm going to post it separately. So be sure and watch the Amoeba Sisters video. And that ends the notes for Unit 9, Part 3.